You have read nucleic acid during your HS class, some part, but with that amount of knowledge, actually you cannot learn the basic biochemistry or molecular biology or biochemistry part. And of course, if you do not understand that thing, then the basis of normal biochemical functions cannot be understood. And until and unless that normal biochemical functioning is disturbed somehow or other, pathological process will not occur and disease will not happen. So it is fundamental to understand this thing. So I will go a little hurriedly because see, biomedical importance needless to say that without DNA, RNA, life is not possible. Nothing, no protein will be produced. So that nucleic acid maintenance structure is very much important for production of proteins, and proteins are of diverse natures including enzymes, and they perform the metabolic functions. And there is an ordered sequence in the DNA which you know that codons are there which code for a particular protein. The central dogma of genetics is the flow of information from DNA to RNA and then from RNA to protein formation final. That is the central dogma. It's a too complex process. The more you learn about say DNA to DNA formation, that is replication or transcription and translation, the more you read, the more you realize your emptiness of understanding. And it is a very, very complicated process, and you will believe after the end of the first year that the human cell is the largest laboratory perhaps of the nature. No other laboratory in the anywhere can function so well coordinated that a cell functions to maintain its structure, to maintain its progeny also. So as I told yesterday in the first class regarding this connection of the two adjacent nucleotides by phosphodiester bond and the stability, thermodynamic stability of the phosphodiester bond. See, these two bases are joined together. Again, I am repeating that this is a phosphodiester bond. This is very important, that may be asked during examination. So DNA contains 4 distinct nucleotides, as you know, say ATGC, and I told you the direction, there is a directionality polarity from 5' to 3' direction in a one strand, and all nucleic acid grow during replication. You will see it will be from 5' to 3'. So that information is there in the ordered sequence, and if there is any breach in the continuity or in the maintenance or actual finality of replication, then that abnormal protein will occur if it is really in a very particular important position. So this Chargaff look you do not know. Again, please stop taking photographs. Everything is written in the book. I am telling you, listen simply. Otherwise, I will be irritated. I will stop the class here. You will get this video in the video from the YouTube. What is the problem? I am there. You can get all. Sit down here. You can get everything whatever I am teaching to you. What I will do with all these things. This for you only. Listen as much as you can note down. These are available in the books. I am telling you, if you cannot understand, then only you will again scroll this thing. The entire thing you will get. You stop this thing and understand. And if you do not understand, I will be very much happy to take another class in small group discussion. You will get 2 hours time. You read, and if you do not understand whatever I will be saying, the entire slide will be shown again. And each and every slide I will discuss to you. If anybody is not satisfied, I will try to clarify with the best of my ability, sincerity and diligence until you understand. That will be my effort. I will take 2, 3, 4 SGD classes. I don't mind, because I will get 2 hours time. There is 1 hour time. Don't disturb me. Otherwise, there is a flow of thought that will be disturbed. Photograph will not take any purpose. You can take multiple photographs. I am having a number of presentations. I get irritated. Concentrate here. The Chargaff rules is very simple. It is A to G, A, T and G, C. We all know that A is equal to T and G is equal to C. So this will be in equal concentration. That was the first rule of Chargaff. So it was in 1940. The first rule holds that the in double-stranded DNA, the AT is equivalent to in equal concentration, equal percentage. And based on that, in 1953, Watson Crick told that double helix model of DNA, which you have seen, that was created. The second rule, which the Chargaff said, that A is almost equivalent to T and G is almost equivalent to C in each equal strand. Remember, it is not in double strand. In each equal strand, it is almost equal. So the complementary strand will have the same thing, but this will not, in a single strand, you will not get all the AT will equal amount of AT and GC. The sequence will not be maintained, but it will be near. So global feature of a single base strand is that it forms that thing that A percentage will be almost equivalent, near. Something is 53, so something will be 49. So G is something, suppose 61, C might be in that particular chain, C might be 65 or it might be 55, that just like that. So why a DNA is packaged, why this ATGC base pair, I am telling you, why this ATGC base pair, why double-stranded helix, why it is required at all? Because DNA would have remained in the single linear strand. What was the requirement of having a double helix strand? Then supercoiling, coiling, supercoiling and compaction. Why? Because if you see all the human DNA, if you collect it from a single cell or in the entire body cell, the length of DNA will be 41 times more than the distance from Earth to Sun. And that we'll see in a little while. I have just collected the actual paper from the BMC Research Notes 2019. You see the size of the smallest cell of the human is 4 micron, that is sperm. And largest cell is, all you remain there, that will be very good. You will not be able to see all these things. The largest cell is ovum, 120 micron. So can you remember in this 4 micron structure, that entire genetic material is stored and during meiosis, it is equally halved. So average required cell size is 10 to 100 micron. So total length of DNA in average human cell, nuclear DNA. Apart from nucleus, in the mitochondria there is one DNA is also there. The size of that mitochondrial DNA is also there. That is not taken into this calculation. So it is basically the DNA of the nucleus. So total length of average male and female is 206 centimeter. So that means 2.006 meter. 1 centimeter is 10 to the power 5 micron. Male nuclear gamete is having 6.2 giga base pairs. See, all you know about gigabyte, mobile. So that is basically, I have converted it just to, why I have converted it? Just to make it into a centimeter. So 6,27,000 lakhs base pair. In females, it is 208. So average is approximately coming 206 centimeter. Now, Sun to Earth distance is 150 million kilometers. That is 149 into 10 to the power 9 meters. So total number of nucleated cells in a reference human being is 3 into 10 to the power 12. So total length of DNA, average male and female, that is 206. So total length of DNA in a human being will be, this will be multiplied, this number will be multiplied into that number, total human cell. So that comes to this much distance. There is 6018 in 6 into 10 to the power 9 meter. Now here the Earth to the Sun distance is 149. So if you divide it, it becomes 40.38. So total length of human DNA will be nearly 41 times than the Earth. So that much nucleus, see, such a huge distance of nucleus needs to be, accommodated in a cell, and that needs to be replicated also. So there must be a mechanism that its structure is maintained, there is no mutation, and it is properly regulated. So several proteins are also present around that double stranded nucleus to regulate its function, when it will be transcribed to form mRNA, when it will be transcribed to when it will be replicated. So this is the reason why I have told, see now this double helix, why ATGC? See, there is a space constraint. In that small amount of space in the DNA, that entire thing has to be packed together, and there should not be any spatial disturbance. Everything is mathematics here. You see the width of the diameter, that is width of this helix is 21 angstrom, this is fixed. This distance between the two helix is 34 angstrom, this is also fixed. Now base per turn, this is also fixed, 10 base per turn. So basically pairing rule is that you see here there is a one, this is called major group, and this small area is called minor group. Here the proteins actually get attached and they regulate the DNA structure regarding its various functions, when it will remain quiescent, when it will undergo replication, when it will do transcription. So pairing of DNA, one purine with one pyrimidine, because there is a diameter of only 21 angstrom in this space, and there is only, if it wants down, there is 11 angstrom space for one nitrogenous base. So here only 3 total purine and pyrimidine will hold, not more than that. So purine, 2 purine if you attach side by side, like A coupling with C, it will occupy more space. And if 2 pyrimidines, then it will be also less, space will be less. So this is a simple mathematics that is followed. If you see biochemistry, go a little higher up, it will be biophysics, and if you go still higher up, there will be mathematics. So this mathematical actual calculation you are seeing, that is the reason why AAT and GC pairing is done. So between A and T, always remember that they also knew that there are 2 hydrogen bonds. So the hydrogen bond forms because between one electronegative and one electropositive atoms. See, GC base pair is stronger than AT because there is a 3 hydrogen bonds, and GC sequence is more resistant to transportation because there are 3 hydrogen bonds, so more energy will be required, more temperature will be required. So average GC content in every per cell is about 40.919%, is almost 41%, is almost fixed for every cell. Now, can't we have, why AT and GC pair, not AG and T? It can happen to purine and as I told you, the 2 purines will occupy 2 more space, 2 pyrimidine will occupy 2 more space, 2 less space. Now, A and C and G pairing is possible, definitely possible. But then that space will not be there. So that hydrogen bonding which is occurring between the 2 will be on the opposite side, and it will take more space. And that is the reason why AC adenine cannot couple with another purine, or this pyrimidine will not form another pyrimidine. See, in that small gap, it is so tightly packed that only in that helical structure, the 3 bases can be accommodated. So it is possible structurally, definitely, but not spatially, it will not allow. That much I want to highlight. So this double structure is there, the helix diameter, as I told you, it is a 10 angstrom, a radius because it is 20 angstrom, and mean weight of DNA in diploid cell is 4 picogram, that is 10 to the power minus 12, and total genome weight is about 19.39 gram. Now you note down this thing. There are 9 points. You note down. See, one question might be given, describe the double helix of DNA. So far what I have told, you will be able to write down. Now just don't forget these points only. These are easy to remember. The measurements I have said. Number 1 point is measurement width 21, distance between the one helix is 34 angstrom, that I have discussed. One base pair turn, 10 base pairs. Bonds between 2 hydrogen bonds between purine and pyrimidine, and between stacked base pairs. One base is one after another, and there is no hydrogen bonding between the two. There are van der Waals force and hydrophobic interaction. During protein structure, you remember that these 3 forces are there, only hydrogen bond, hydrophobic interaction and van der Waals force. You just read from the physics what is all these things, so that if anyone asks, you will understand. So these are the 3 things. Well, electrostatic bond you know very well, hydrogen bond, then hydrophobic interaction and van der Waals force. You read from any physics book or from internet, you will get an idea. So between 2 strands, remember, hydrogen bond, and between bases in the same stack, it is hydrophobic interaction and van der Waals force. So pairing base pair, it follows Watson-Crick. Viscosity, remember, this is one point I want to highlight. In native state, means original state, it's rod-like and it's highly viscous. So it is extremely viscous due to combined base stacking and hydrogen bonding. Polarity, one strand is moving in 3' 5' direction, one strand is moving 5' to 3' direction. So one is parallel, this polarity is there. Groups, I have shown, that is major group and minor groups. These are the sites for attachment of proteins for regulation of DNA. And winding parallel to phosphodiester bond, remember this phosphodiester bond. হয়েছে? একটু তাড়াতাড়ি লেখো, 2-3টে জায়গা লিখতে বলবো খালি, আর কিছু নয়, বাকিগুলো পড়ে নিও। Strands, this you note down, this you try to understand first. See, here you understand. This part is there in the book, the only understanding part is required. See, in DNA, one strand is particular template strand. For your understanding, I am telling you, the gene resides in the template strand, not in the coding strand. Opposite strand is coding strand. Why I am telling you? See, if you recall the codon is not as per sequence of DNA. I am telling you, see, this is the genetic code. Is it as per sequence of DNA or RNA? How many is in favor of DNA? Whether the codons are as per structure of the DNA or as per structure of the RNA. You should try to understand. This is there a little bit understanding here. Codons are as per DNA or as per RNA? All of you are sure. Why RNA? Why RNA? Just one question, why it is as per RNA, not as per DNA? Bolo? Bolo? Jure bolo? Yes, uracil. Correct answer. এতজন রয়েছে, কেউ বলতে হলো না? প্রথমে তো বলে দিলাম যে ডিএনএ নয়, আরএনএ। কেন আরএনএ? বিকজ এইখানে দেখো, তাকিয়ে দেখো, জাস্ট লুক এট দিস থিং, all the structures are U, not T. So codons are in the form as per structure of the mRNA, not as per DNA. Now you note down in this 5 point, AUG is going to be methionine. On your paper you note down. AUG is methionine, THR, ACC representing THR, threonine, AA is lysine, and AGC is arginine. Just note down. I'll clarify that thing. So codons are as per mRNA, not as per DNA. Now, why we said at a coding strand? Try to understand this part. Genes are not located in coding strand. Genes are located in template strand. Why? See, now you see here, see this is the RNA transcript, that is mRNA. One is 5' end, one is 3' end, and there is U here. Do you get my point? This is U is here. Now this is coding strand as I told you, and this is template strand. Template strand, one strand is produced. So this is complementary. Do you get my point? This is complementary. So if you look at this primary structure, the primary transcript, that is actually forming the codon, is matching with the coding strand, not with the template strand. But it is basically a copy of the template strand. Since it is matching with the primary transcript, is matching with the coding strand, so these are called codons. Codons are there as per that sequence. Normally we say. So here, as you noted down, just look at the structure. See, this is the RNA structure, 5' end and 3' end. So AGG is arginine, AA is lysine. Then it is chain is growing, and ACC is threonine, and AUG is methionine. Do you get my point? So one information, the codons are as per sequence of the RNA, mRNA, and the mRNA strand, that transcript matches with the coding strand of the DNA. Is this point clear? So here you see the pairing occurs here. The mRNA strand, this is that codon basically, and is pairing with the tRNA. So opposite strand is coding strand because it matches the sequence of RNA transcript as I told you. So coding U instead of T, that's the difference. So this is again occurring between 5' to 3' direction. This is in this direction the DNA replication is taking place. So proteins in the groups. This you will get in the book. Just as I told you, it interacts with the DNA with the exposed part of the nucleus, because proteins will not be able to go inside. It will be available on the outside of the DNA structure. So they recognize and bind to the specific nucleotide sequence. There will be multiple proteins. Histones, you have heard the name of histones, and that gives an unique shape. It contributes critically to cellular function. As I have been telling you, these are regulatory proteins. They are involved in DNA replication, repair, recombination, and transcription of the genes, because when protein synthesis will be required, mRNA will be produced. So base modification, I told in the previous class, all the bases which you know, they undergo extensive modifications, and methylation at one point I told, highlighted. So you can see bases are extensively methylated. There is 6-methyl cytosine, 6-methyl adenosine. So what was the role of methylation? It basically, remember, just like in this metabolism class, you will say some reactions, some enzymes get activated on phosphorylation, some get on dephosphorylation. Similarly, methylation condenses and represses activity, whereas demethylation leads to transcriptional activation. And methylation also identifies which one is the parent strand from where it is copied. And one important point I told, that paternally derived allele is methylated. Methylation study is done. So DNA strands during replication or during transcription it separates. The same thing can be done in the test tube in the PCR reaction. So DNA can be reversibly denatured and also it can be renatured. So separation of double-stranded DNA into single-stranded DNA is called denaturation, and denaturation can be obtained by in the test tube by increasing the temperature, by increasing the salt, by minimizing the salt concentration, or adding cheotropic agents which will basically disrupt the hydrogen bonding. It can occur either alone or in combination. If you give high temperature, then the strand will break. So it will form single strand. So renaturation, again, if you decrease the temperature, and that is what is the principle of PCR. Once we denature the DNA, get the double-stranded separated, allow the extension to occur, and then after this thing is completed, decrease the temperature and allow it to reanneal. So this reionization is highly specific because there will be strand complementarity. In the living cells, it occurs naturally. As I told you, DNA replication. Replication will occur, one copy will be formed, and that will form again the daughter cell. And in the daughter cell, that copy will be there. So DNA recombination, again, DNA repair, DNA gene transcription. Hyperchromicity is one part. In the first class, I told that purines and pyrimids, they basically absorb UV lights at 260 nanometer. So if the DNA strands remain double-stranded, so its optical absorbance at 260 nanometer will be lower. If you denature it, there will be increase in the chromicity, that absorbance, that is called chromicity. Once we denature the DNA, its chromicity will be increased, that is called denaturation. So as we increase the temperature, the DNA strands will progressively separate. So progressive increase will be there, as you can see here. See, this TM, note down what is TM, this can be asked during examination. See, each DNA will strand over a particular range of temperature based on its GC content. So the midpoint of the measured DNA denaturation is called TM. See, here it is the temperature is low, temperature is high. Now we have started increasing the temperature, there is a 50%, this is the TM. Here in this side, the DNA is double stranded, and here the DNA is 50% separated, and if you increase the temperature further, there will be two strands will be totally separated, and normally if we rise the temperature more than 80 degree temperature, the DNA strands will separate. Again, of course, based on the GC content. The more the GC content, a little more temperature will be required. रेजिन একটু ইয়ে করে দেখো তো, প্রেস করো তো, নিচেটা প্রেস করো, ডাউনটা প্রেস করো। হ্যাঁ, factors affecting DNA, as I told you, if the GC content is high, then that will increase. Now, phosphodiester bond, there is a phosphate bond, phosphate is negative charge. So DNA is normally negatively charged. So if we add positive ion to it, so that charge will be neutralized. So if you add a 10-fold increase of monovalent cation concentration, that will neutralize the negative charge, and it will increase the TM value by 16%. See here, the first curve is a high GC content, as I told you, because it is having 3 bonds, and this is having a low GC content, that's why its TM is lower. The forms of DNA, just remember this thing. From A to E and Z, there are 6 forms which are available, and these are basically due to interstrand composition. What is the basis of these 6 forms? And that is structural rearrangement. In nature, only 3 forms are found. That is A, B, and Z form, and B form is found normally under physiological conditions. So this is the common forms in the nature. This is based on the X-ray crystallography. Again, all the structures in biochemistry were done basically again by using physics, that is X-ray crystallography. So these are the basically schematic diagram of different forms of DNA. So the common form are A, B and Z form. See helix. Helix, it is a right-handed helix. Why? How will you identify it is a right-handed helix? You put your thumbs up and the other fingers down. This is the way, this is the axis of DNA. And this is the actually basically that DNA double helical strand, these forms. So that strand, you see, look at this thing, it is clockwise, it is right-handed. Now if I put this thing, it is left-handed, it will be anti-clockwise. So it is, this is the axis of the DNA, and remember this is the actual, the double-stranded structure. That is a simple thing. So there is an axis in the double helix, and the DNA is moving like this in a right-handed helix. So I told this thing, the base pair turn in the B form is common. So 10 base pairs per turn, and there is a helical diameter is almost 21 angstrom that you have noted. So types of DNA structure, just like protein, there are 4 forms. One is primary, secondary, tertiary and quaternary. So primary, just like protein again, it's a linear sequence of polynucleotides that will form the linear structure, primary structure. And based on that primary structure, again the secondary structure, tertiary structure, everything will depend on that primary structure. So somebody was asking one day on the other day in the class also that regarding the functioning of pyrimidine analog. What is the function? Then I explained to her in person in the class that if you see if there is proper pairing is not done, that if instead of uracil, 5-fluorouracil is incorporated, then the primary structure will be disturbed, and that will not allow the DNA to fold properly. So primary structure, secondary structure, it basically binds to each other, and the double helix structure which you see is essentially a secondary structure. And where that 3 things I told, hydrogen bonding between the 2 strands, hydrophobic interaction of van der Waals force between the stacked bases of the adjacent in the same strand. And it's because of the structure, because of the content, it shows a high amount of structural variability. So secondary structures of RNA. RNA is not double stranded. RNA at some point of time gets folds. There are 4 things which happens: helix, bulge, loops and junctions. So where there is a turn, where there is there are 2 strands, usually bonding occurs. And this bonding also follows that base pairing rule. One is moving in the 3' 5' direction, one is 5' 3' direction. That ATGC rule will be there only where there will be 2 strands will be running parallel. So you can get a bulge, you can get linear forms, you can get hairpin loop also. And this structure is important during RNA structure, that you will see shortly. So you will see a hairpin loop. This you will see in the clover-leaf structure of tRNA. You can see stem loop, you can say pseudo knot. There are 2 loops, one is covering the 50% of the other. So tertiary structure is a higher order structure. It is still compact. Now it is still compacting. It is again just like 2 ropes which have been wound together, and now you are finally just like woolen ball. You are trying to do that thing. So further coiling is done, and there is large scale folding, and it gives the 3D space, 3D actually shape. And it is this 3D step on which these proteins attached, and this is responsible for actual functioning of that functional DNA because of the tertiary structure. So that right-handedness, left-handedness are explained. And length per helix turn you have already understood. Number of base pairs have discussed. Difference in size between major group and minor group where it is located, I have said. Quaternary structure. When this structure, to this structure proteins are attached, that is called quaternary structure. See, supercoiled and relaxed form of DNA. See, when the DNA is in the circular form, you see, so if you twist that circle, it will assume a position like this figure up 8. If you twist further, it will be like this. So it will put torsional strain. So the more you twist it, the more it will be compact. So DNA is wanted in supercoiled form, so that the entire space, whatever available within the nucleus, will be utilized fully. Okay, there should not be any gap, and that is the reason why supercoiling is required. So again, if you do the reverse thing, then uncoiling will occur, and that happens. What exactly happens during replication? Because in that form, replication cannot take place. For replication to take place, we need a linear strand, so that needs to be again reversed. So during packing, we are doing one thing, that coiling and then supercoiling, and when you are requiring replication or transcription, you are doing just the opposite things. So there are two enzymes. One is supercoiling, one is coiling this thing, one is uncoiling this thing. So gyrase is the enzyme which coils. And topoisomerase, remember, just note down, gyrase is the enzyme which inserts supercoils. And topoisomerase is the enzyme which just do the reverse thing. So that relaxes the supercoils. That you will be taught during your that replication class or transcription class. What are this is supercoiling. See, there is one cylindrical structure. This is the rupal structure. Then you just go on another protein. If you go on wrapping like this will call a supercoil structure. Now, if you undo this thing, if you remove that thing, that protein part, it will uncoil. And supercoil form is the preferred form. As I told you, it will occupy less space, and then it will be more compact form. That structure will remain in more compact form. So remember, only in the entire DNA part is not useful, just like our human population. Only in any specific area you see a few persons remain active, other remain as silent spectators. Same thing is here. So that means other persons are not doing anything, they are doing the regulatory jobs. They are not simply idle. Those who are watching and looking or hearing something are not doing anything that is not true. Some part is doing the activity, somebody is there to observe it, somebody is criticizing, somebody is regulating their activities. The same thing is true here. Only 10% of the human DNA contains gene, and that is the active part, what we call. Just like our political system, you see there are only 541 MPs, they are regulating the India. From them the ministers are selected, and they are taking the decisions of the entire populations. And in any area you see, there are a handful numbers of persons which are active. But to support them, there are many things. The introns are the places where basically the proteins, regulatory regions are there, regulatory proteins act, and they control their genetic activities. One leader cannot function without supporters. Not possible. The leader cannot be leader without a follower. Simply a gene cannot function in that particular gene cannot function in isolation. See here, what is happened during primary transcription. So there are intervening areas. So these are the coding areas. These coding areas are called exons. Note down. The coding areas of the genes are called exons, and the intervening areas between the exons are called introns. Introns do not take part in actual final protein translation. When DNA is transcribed to RNA, the entire thing is copied. That is exon, intron, everything is copied. But from when a mature RNA is produced, from that these particular introns are spliced and only the exons are joined together. That is called splicing. So some amount of endonuclease activity must be there. So that endonuclease will cleave the bonds which is present within that DNA strand. So that is called mature mRNA. Once this mature RNA is formed from the nucleus to the cytoplasm. So all the transcriptional modification takes place within the nucleus. So this is a higher level of DNA organization. So the DNA is organized within the nucleus, and the double-stranded DNA is wounded over histones, as I told you, that forms nucleosome, and long stretches of DNA in association with histone forms chromatin, and condensed chromatin forms chromosome, As I told you in the beginning, we know that we are having chromosomes within the nucleus, but in human DNA or any other DNA, there are other eukaryotes, mitochondrial DNA is also present, and they are having separate genes, they are having separate protein synthetic mechanism. Remember, we all inherit our mitochondrial DNA from our mother. Mitochondrial DNA is exclusively transmitted only from the mother. So, any diseases associated with mitochondrial DNA will be basically inherited from the mother. Remember, mitochondrial DNA inheritance. In other cases, we get one copy from father, one copy from mother for a biological child. But mitochondrial DNA is totally inherited from the mother. So, that is an extra chromosomal DNA. Similarly, in the bacteria, we have extra chromosomal DNA, that is plasmid. So, DNA wraps around the histone, as I told you, as we form, increase the form, this will finally see the, this you just remember, all these points you note down. Rest you know, what you have to note down is just for sugar, RNA, maybe a very easy question, write down the differences between DNA and RNA. So, just like that double-stranded DNA characteristics, note down the points, sugar, deoxyribose, ribose, bases, that uracil and thymine, strands double-stranded, RNA can be double-stranded also. As I showed you, there will be opposite strands where it can form a base pairing. Again, in the tRNA structure, I will show it to you. So, RNA can have double-stranded structure. So, ATGC content is equal always in DNA, but ATGC content is not equal because it is a single stranded. Occasionally, there is folding. So, stability and hydrolysis. As I told you, DNA is extremely stable. Last time, third amino stability I said, but RNA is not that stable because why? In 2' position, there is a hydroxyl group, and that is the reason why RNA is not stable. Content of DNA almost absolutely constant from cell to cell, but RNA content varies because the protein synthesis does not take place in all the cells. Liver is very much active in metabolism, but RBC is not that active. It does not have mitochondria. So, all cells are not equally active. So, therefore, the content of mRNA will not be same. RNA will not be same. Lifetime DNA is longer, lifetime DNA is short. Catalytic activity, yes. There is no catalytic activity of DNA. RNA is a catalyst, can act as an enzyme. Normally, we all know that all enzymes are protein in nature, exception is tRNA. Why tRNA is a catalyst? The last slide is there to note down the points. If there may be a single question, explain why RNA is how RNA acts as an enzyme. So, presence, of course, RNA is present in the RNA, DNA, everything is present in the nucleus, nucleolus, but RNA is specifically present in ribosome and cytosol. Mitochondrial DNA is there, mitochondria is part of cytoplasm. And in plants, chloroplast, it is not our business. Remember, mitochondria contains DNA and we inherit the mitochondrial DNA from our mother. Classes and types of RNA, you know these 3 types, main types, mRNA, tRNA, RNA. Just note down one thing, small RNA. And there are many other types of RNA. I will discuss only these five types because you are no longer class 12 student. So, this mRNA, tRNA, RNA is not sufficient at this stage. You need to add something more. So, there is small RNA. So, there are non-coding and coding area, heterogeneous nuclear RNA. What is heterogeneous nuclear RNA? That primary transcript which is formed from DNA. So, this is heterogeneous in the sense it contains exon, it contains introns, it contains other regulatory steps. These all are removed. So, this is heterogeneous in structure, and in the nucleus all these structures will be found. So, that is heterogeneous nuclear, that is a precursor mRNA. Just like protein, a precursor protein is there from which the actual protein is formed. So, that precursor protein. So, these are unmodified RNA molecules synthesized from the template DNA. So small nuclear DNA. Now you note down here the catalytic activity of RNA. Small nuclear activity, snRNA function as splicing and endonuclease activity. As I told you that during that primary transcript to actually mature RNA, endonuclease activity must be there because that intron part has to be removed, All the introns have to be removed. So that is done by the splicing, and this is done by the snRNA. It is having an endonuclease activity. MicroRNA and small or silencing RNA, they are inhibitors of gene expression. So gene needs to be regulated, and this uncontrolled gene expression occurs in cancer cells. It doesn't occur in normal individual. Because if there is no genetic control, gene expression control, the cells will undergo huge proliferations and there will be tumor, and if you still not control this thing, they will lead to cancer. So snRNA, just note down this thing, they need not read this thing, only note down U1 to U6. This is the part basically which is involved in splicing business, that is catalytic activity. So function is, it's a pivotal role in processing RNA, particularly mRNA, from the mature primary transcript to mature RNA. It is present, yes, it is present in nucleoplasm, nucleus, and you see nucleus and cytoplasm in both the places. So we get viral infections, RNA. RNA, how does it integrate into the human being and take control? Just like East India Company. Just like East India Company, the DNA virus or RNA virus gets integrated into our system. RNA viruses, actually through retroviruses, they have a built-in viral RNA-dependent DNA polymerase. So from a single strand, they make a double copy. So they become double copy. Suppose here, you see human being are having a 1 and 2 gene, we are having on the chromosome. Now this is the viral gene ABC. What it does, it basically comments here, and then it tries to integrate. Now you see the 1 gene number gene is here, that 1 number gene is there, and now 2 number gene was there in the human gene, and this 2 number gene is there. In the middle, the virus has incorporated its genome into the host cell, and now it is taking full control of the host cell, and it will undergo replication. That's why viral load becomes very high, and cell will no longer be able to perform its normal function because its function has been taken over by that infecting agents. So that is the mechanism how viral DNS in the early stage of infection, viral infection, if you give the antiviral drugs, the infection is under control. But when it has happened to a great extent, it will be very difficult. So mRNA, basically 2 to 5% of the total eukaryotic cellular RNA. A messenger RNA, as you know, that carries the actual information from the DNA to RNA, and it is the most heterogeneous because too many proteins are there. So the mRNA will not be a simple thing. So it is, there will be both transcriptional and post-transcriptional modifications will be there. One important point, note down 5 terminal of the mRNA, note down here. It is capped by 5-methyl guanosine triphosphate. Note down this point only. 5-methyl capping, 5' capping is done by 7-methyl guanosine triphosphate, and its role is basically to transport from nucleus to cytoplasm and promote binding of RNA. And this is the 5' terminus which starts giving signal the protein synthetic machinery to start protein synthesis. And again, this structure 5' end is supposed to get inactivated by 5' exonuclease activity. So 5' exonuclease activity is there in our system. That 5' exonuclease activity can destroy that 5' end of the RNA, mRNA. So this 7-methyl guanosine cap protects the RNA from 5' nucleoside activity, exonuclease activity. Similarly, 3' end is kept done by the poly A tail, the polyadenosine tail, that also prevents that mature mRNA to be destroyed by the 3' exonuclease. Both 3' exonuclease and 5' exonuclease is there in our cell, but somehow that mature mRNA which has been produced after so much effort from the DNA to mRNA, that must be protected from being destroyed by its own system, so that the protein translation can take place. So for that 5' capping and 3' capping by the 7-methyl guanosine cap and 3' poly A tail is there. So this is the 5' cap. So there is, you see, there is a 5' untranslated region is there, but actual protein coding sequence is there. So that part is preserved. So similarly, 3' end is poly A tail. So you see the primary RNA transcript, as I told you, is exon, intron, exon 2, intron, exon 3. Now RNA processing has taken place within the nucleus, and finally what we got, exon 1, exon 2, exon 3, and at one end the 5' end, the 5' capping is there with 7-methyl guanosine, and 3' end is poly A tail. So these, as I told you, this primary processing, that heterogeneous nuclear RNA part takes place within the nucleus, and once it is matured, then it is exported to the cytosol, and in its association with ribosome, it starts formation of protein translation with the help of tRNA. The post-translational modifications I have told, that is basically both cap, that 5' capping, and poly A tail addition. So it occurs in the nucleus, and after nuclear processing is complete, then only it comes into the cytosol. I have been repeatedly telling, I told us 10 times that the primary processing has taken place within the nucleus. After this mature RNA is formed, then only it will be transported into the cytosol for translation to occur. So clover leaf RNA structure is a common question. What is a clover leaf? How does it look like? A clover leaf looks like this, and in our system the Susuni shark is very common in villages. It looks like that. So that triple helical, এটাকে আবার ত্রিপত্র বলা হয়, এটাকে বা বাংলায় বলা চারপত্র পাওয়া যায়। তো এই tRNA স্ট্রাকচারটা, just look at this thing. This is a normal structure. I have just inverted the structure. So it looks like that clover leaf. So the name of tRNA structure is clover leaf because of the structural similarity. So tRNA are 20 times because there are 20 amino acids. Simple. So again it is having a primary structure during that thing I have described. So it is having, there is extensive folding, and because of that it assumes a structure of clover leaf. There are 4 arms of tRNA. Just note down. One is 3' end acceptor arm which accepts the amino acids. Anticodon arm is second one, this one. It interacts with the mRNA, and that corresponding amino acid is attached here. DR, it is the recognition site of amino acid tRNA synthesis. DR means dihydrouridine or dihydrouracil. T pseudouridine arm is here. It fine tunes and stabilizes the regional structure. This extra arm which you are seeing, it is a variable arm. It recognizes mRNA molecule. It is not present in all the cases. So this is the structure. Again, I am telling you. This is one is acceptor arm, one is D loop, one is T pseudouridine loop, and one is anticodon loop. There are 4 structures you must remember. 4 arms. One is this acceptor arm, that is CC arm, which is coupled to a hydroxyl group. This D arm or D loop, and T pseudouridine loop, and anticodon loop. নিচেটা দাও, এইটা দেখো, আর আগেরটা যাও, এইটা দেখো, এটা তো বললাম, পরেরটা দাও, এই দেখো প্রাইমারি স্ট্রাকচারটা, ইটস এ 76 nucleotide primary transcript. This is the secondary structure of tRNA. You see here this U is modified to D. This is dihydrouridine, and U to U7, 16 and U17 to D16 and D17. This is basically dihydrouridine. This D is representing dihydrouridine. So this is anticodon loop, and this is this T pseudohydrogen is here. Sorry, এখানে পিকচারটা এখানে চলে এসেছে। এইখানে তাকাও বেস্ট look at the structure here. It is U54 and D55. This is U is converted to pseudohydrogen. So this is finally this tertiary structure. So it is a cloverleaf structure in the secondary form. When it is in the tertiary structure, that it takes a L-shaped structure. পরেরটা দেখো চলে এসছে অলমোস্ট এন্ডে চলে এসছে। So this is amino acid moiety. As I told repeatedly, this is helping in recognition of amino acid tRNA synthesis. This variable loop, you see in the variable loop, you look at the structure very closely. M2G means 2-methyl guanosine. D means dihydrouridine, as I told you, D loop, and this is pseudouridine loop, and this is M to the power 7G means 7-methyl guanosine. So ribosome and protein synthesis, this is the applied aspect. Antibiotic use, how antibiotics works. Just note down this thing. This mammalian ribosome has 2 subunits, human mammalian ribosome. One is 60S, one is 40S. Note down. One is 50S, one is 40S, and ribosome acts as a machinery for protein synthesis. And when the mRNA combines with multiple ribosomes, that is called polysome. And it is the polysome which forms the protein. Once the protein synthesis is complete, then the 2 subunits of ribosome is separated, and that mature protein molecule is released. So the basic purpose why I am telling you, again I have just this slide I'll show you the difference. So components of mammalian ribosome is 40S subunit is formed by 18S and 30 polypeptides. See, if you look at the RNA structure, it is having 5S, 5.8, 18 and 28S. S is basically Schaffberg unit. You will be doing the centrifugation in your laboratory. If there is not done, I'll demonstrate it. If you have a suppose turbid solution, if you allow it to settle, it will definitely settle. It will take some time, but that can be obtained at a faster rate by rotating using a centrifugal force. So it is a sedimentation coefficient based on the molecular size. So there is a 40S subunit, and 40S subunit is having 18 subunits plus 30 polypeptides, and 60S subunit is having this RNA molecule and more than 50 polypeptides. It is 70% of the total cellular RNA is basically because of tRNA, because so many amino acids has been brought. This is the actual applied aspect. See, eukaryotic ribosome, this is our ribosome is 8TS ribosome. It is composed of 2 units, one is 60S, one is 40S. 60S unit is composed, just now I showed 40S and 60S and 40S. Prokaryotic ribosome is having 70S. It is composed of 2 units, one is 50S, one is 30S. Don't try to add together, it will not get the figure because there are 2 different subunits that are having different shape, different size. So they will have a different sedimentation coefficient. So it will have here it is 50 polypeptides, you see here it is 34 polypeptides. 40S it is 18 polypeptides, 30 polypeptides, this is 21 polypeptides. Because the association of this 70S and 30S as the target points for antibiotics, we will give antibiotic which will destroy the bacteria or virus, not the human DNA. So we have to specifically design an antibiotic which will not destroy the eukaryotic ribosome units aggregation at all, but it will not allow this prokaryotic ribosome. That you will read after protein translation, and then how these translations are inhibited by using that antibiotic. Yesterday one of your senior student told me, sir, I could not understand pharmacology. Sir, mechanism of action I am not understanding, because I told you that if you don't know biochemistry, it is not possible. So what is pharmacology? You are interrupting either a biochemical basically pathway in some way or other. Either you are modifying, either you are increasing or you are stopping, allowing it the reaction to occur at a faster rate, or you are simply stopping. So here if these prokaryotic ribosome are not allowed to associate, that prokaryote which has infected our system will no longer be able to survive, it will die. So this snRNA, what I told, that U1 to U2 is function, they are basically spliceosomes. So RNA acts as a ribosome, just like note down this thing, this is the last slide. RNA acts as an enzyme, it is having catalytic activity, it is having 4 activities. One is phosphodiester cleavage, bond cleavage. Post-transcriptional splicing and endoribonuclease, I have repeatedly told. snRNA is involved, snRNA is involved with E1 to E6. Amino acid hydrolysis, this you will read during protein synthesis, peptidyl transferase activity is also there in the RNA, and transesterification. These are the 4 enzymatic activities which are present in RNA. So RNA can act as an enzyme, so it is called ribosome. এরপর দেখো small RNA, just 3টে কথা বলে, this is the last slide again. Small RNA is one form, I told you, it forms 5% of the total eukaryotic RNA, and specific nucleases are there to digest nucleic acid. One is deoxyribonuclease, it basically clips the DNA, ribonuclease, RNA, an enzyme capable of hydrolyzing single or double stranded bond, exonuclease. Basically clip that 5' exonuclease will clip the 5' end. 5' end exonuclease, 3' exonuclease, one end will be basically hydrolyzed, but endonuclease will clip the internal bond between the in the nucleic acid. So during recombination study, you will see the restriction endonuclease. Restriction means there are restriction sites. A specific sequence of DNA will be there, and restriction enzymes will cut only if that sequence is present, and it will cause restriction of fragment length polymorphism. If we allow our DNA to be cut by one restriction nuclease, we will get suppose 5 fragments. Now if we cut it with another fragment, it will give 10 fragments. If we cut it by third one, it might give 3 fragments, and there will be overlapping of these fragments, and using that overlapping, actually the entire sequence of the gene is determined, that you will learn during DNA sequencing. So with this I conclude. I think you are today bored. This is a long class, I am helpless, because without that thing you will not understand this protein translation transcription. So read it very carefully, and if you require this class to be repeated, I will be very much happy to take the previous class and also this class as many times as you want. As many times as you want. I will not feel tired. I will be happy. Be it here in the lecture class, I will can take it on the, and in a separate, even in the evening class, I can take it. No issue. Okay.